So good afternoon, everybody. I'd like again to thank the organizing committee for extending such a generous invitation to come all the way to present our technology to this forum. Thank you for attending. It's been a long day and you've been exposed to many challenging and many innovative technology. I do hope you have a little bit more span of attention to hear a little bit more about something even more exciting and more I would say thought-provoking than what we've discussed this morning. I'd also like to thank the former presenter, Luke and Professor Nielsen, who discussed synthetic biology, transformation, genetic engineering of plants, of bacteria, and of insects, because Moflora is really going to change that perspective, because up until today, everybody is talking about inheriting a trait by altering the chromosome and creating a new transformed life form. We are offering a different approach. I will share this with you in just a few minutes. It's all in this small sacket. But before we get to that, just a few words about Moflora, the company that I'm being privileged to lead for the past several years. The company is engaged in licensing and commercializing technology that comes from the Hebrew University. The technology that we have called trade-up is a fast way to deliver traits into plants and seed without changing the genetic makeup and without it being inheriting to the next generation. The technology has been recognized by the Agro Plant Protection Forum as the most innovative biotech platform. The company as a company has been honored by Red Herring to be one of the most 100 promising business in the European business section. In the backdrop of Moflora stands the Hebrew University, the biggest university in Israel and actually the largest powerhouse for biotech research and development. Actually, within its several decades of activity, there have been more than 700 registered patents, over 500 different licensing deals, who have created an enormous wealth to their licensors. It is estimated that the annual turnover in terms of revenue from products based on innovation coming from the Hebrew University exceeds $2 billion a year. So quite a substantial experience in taking innovative scientific discoveries right all the way into commercial success in the market, and Moflora is relying significantly on that track record. Maybe I would, in this opportunity, tell you that Israel, that small country you often read about or hear about in the news, has become what is people refer to as a startup nation. It is a vibrant country steaming with innovation and entrepreneurship. We have been pioneering many areas in the life science industry, including medical devices, pharmaceutical drugs, excuse me, agriculture. We've also been very innovative in areas of communication, internet security, and so forth. If you go and look at the companies listed on the NASDAQ, of course, most of them are American. After that, you get the Canadian countries, and after that, you get the Israeli companies. So a lot of the innovation from Israel through the entrepreneurship economy that we've built has been exported overseas in successful companies and successful businesses. The team that I work with have a profound scientific background. I'm privileged to be working with this team. I'd like to mention in this opportunity, since we've mentioned scientific biology and its application, that Professor Ilan Sela, who is one of the co-founders of the technology, was actually the first biomolecular scientist that have managed to transplant a human DNA piece into a plant and have that plant produce actually a human-based protein. Chaim Rabinovich has been the rector of the Hebrew University. He's referred to often as the father of the seed industry of Israel. Is a renowned tomato breeder. He was actually Professor and Nahum Kedar partner when they both set up to make the tomato have a longer shelf life. And up until today, actually, the longer shelf life that tomato growers enjoy and consumers enjoy is mainly due to their groundbreaking discovery. They later on went and bred the cherry tomatoes. Based on those innovations, actually many vegetable companies were established in Israel, later being acquired by international seed companies, and most of this technology is now available worldwide. But actually the most interesting aspect of technology is actually based on a discovery in Professor Ilan Sela's lab. Professor Ilan Sela is a plant virologist who have studied deeply the interaction of viruses and plants, plant viruses and plants. And to give you a little bit of a background, again, up until today, we talked about the need to change the chromosomal structure in order to inherit a trait. Well, actually, if you look into the cells, you would see that there are also DNA particles which are external to the chromosomal DNA. They are often referred to as episomal DNA structures or plasmid. They often appear in this green round shape, and as you see, they are detached and they are not part of the chromosomal DNA. In this way, in nature, for example, foreign DNA such as plant viruses spread in the plants. The interesting thing about external DNA is that plants do know how to recognize foreign DNA and take them through the entire production of protein process as if it was their own DNA. So if we're looking at the process of turning a DNA into transcribing it to RNA and have that RNA produce protein at the ribosomal binding site, there's really no difference between having that in internal and external DNA. And this is a fact that in nature is being used by plant viruses. This is exactly what they do. Plant viruses, as you may well know, they are being transmitted through the saliva of sucking insects. Once they get into the leaves, they actually go into the sap, and from there they spread. Once they go into the cell, they start to produce protein based on this mechanism in the plant, which enables the production of foreign protein. As a result, you will find traits, new traits in the plants, what you would, as farmers, refer to as decay or as rot, but it's actually foreign protein produced by the plant as a result of the viruses producing or presenting external DNA for the production of the plant. Professor Ilan Sela have found that in all plant species, there is a single and generic mechanism to identify this foreign DNA. And Moflora has the license now to use this mechanism to present to the plant beneficial DNA part to induce for a single generation beneficial traits. Let me show you in a short animated video how is that supposed to work.
Quickly deliver traits to plants. Here's how it works. A trait is chosen, such as fungi resistance, virus resistance, or crop enhancement. Seeds are treated with Trade Up. This is a proprietary method that delivers genes using a simple and short seed treatment, not by breeding or genetic engineering. The genes enter some cells, but do not become a part of the chromosomal DNA. As a result, this safe technology does not change the genetic makeup of the plant and will not be passed down to future generations. As the seeds grow into plants, Trade Up is able to replicate itself and move through the plant while carrying the genes with it. Trade Up maintains replication and spreads throughout the plant's foliage and fruit. This process results in reduced dependence on chemical control, reduced pollution, healthier crops, and increased yield.
So this is I would say, the scientific background of what Moflora is engaged with. And as you can imagine, by this stage, this is really a game changer. It's a paradigm shift in the industry. Again, just having a thought that we're not going to clone a trait into a plant. We're going to use a vector, a plasmid vector, to mediate the delivery of the gene into a plant is actually really an exciting vision. I would like to use the vaccination analogy to make this very clear. Today, when you're talking about human vaccination or the vaccination of livestock, you're actually talking about the same thing. You're talking about an external DNA that is introduced either to human or to livestock, which provide them a trait which enables them to negate a symptom or to fight off a disease. And we largely accept and commercially use vaccination in livestock and in men. But up until today, we didn't have this approach to plants. So perhaps in an analogy which is not purely correct scientifically, but is, I would say, comprehensible, we're talking about actually applying vaccination to plants, giving them a genetic material that will enable them to obtain a specific trait through their entire lifespan without having that trait inherited to the next generation. Now, there's, of course, major bottlenecks in the industry that this technology can help solve and, of course, enable many other applications. Let me just reiterate the process a little bit and maybe add some timelines to it so you will understand how much time involves of applying this technology. So the first is, of course, to choose the trait of the sequence. This is not a specific activity that Moflora is engaged with, but I can assure you that across the globe, every year, the big gene discovery companies are scanning anywhere between 70 to over 100,000 different genetic constructs, just in order to isolate and identify which of them is the beneficial genes that we are after when we are solving agriculture's main issues. And I can already share with you that there are dozens of beneficial genes that are being scanned and identified. Only few of them have found themselves already regulated and bred into commercially available seed lines. Most of them are still under R&D, but this knowledge is rapidly expanding. So we have a lot of genes to take advantage of. So once there is a gene there, to clone it into the trait-up vector is a very simple action. It really takes just several weeks. And then at Moflora, we produce enough amount of genetic material sufficient enough to treat the seeds. And this is actually what we put in the sacket here. That's another production which takes probably a week or two. And then we have a sacket which contains dried genetic material. So actually the trait is in the sacket. If you like to think about it, this is a revolutionary way to spread IP. Think about how you got improved quality traits to be imported into Australia. You would have to import overseas traits by buying new lines, by importing seeds. And if you would like to acquire a trait and bring it into your local variety, you would have to put in place a breeding program to breed an external line with the local line to have this desired trait being bred into your local lines. Now, all this can be a matter of bygone past if you could only have the possibility, which now Moflora presents to you, to order the trait by mail. So any new trait available on the globe from any gene discovery company can be, in a matter of weeks, turned into a commercially available powder or active ingredient that can be then added by a seed treatment onto your local seeds. Now, the seed treatment, of course, itself actually is a standard seed treatment. By now, we have proven that our technology can be seamlessly integrated into existing seed treatment protocols. For example, the priming protocols, the wet protocol, which is used to prime vegetable seed, but also to the dressing and coating protocols, which are used to apply chemicals and biochemicals protection over commodity crops, such as the soy and the corn. And once the seed treatment is applied, the trait then expresses itself within the plant. So the entire process, and I'm not talking about the regulation, which will take time, but the entire process is, just a couple of weeks. And let's look at how the process looks like today when we are modifying crops. And you will find out that, first of all, just to find out the desired trait or to identify that specific gene that we are after, whether it's a nematode-resistant genes for soy or drought-tolerant gene for corn, it can take, in average, just 6 years. A lot amount of effort to screen libraries containing thousands and thousands of candidate genes, all being introduced through transformation either to the target crop or to a model crop, and then being phenotyped to address the issue. But once you already identify the genes, you start in a decade-long process of commercially introducing this gene into the marketplace by the use of a breeding program, which then need to be regulated. Now, whether your breeding program is a standard breeding program or whether you are relying on transgenic engineering, it takes you about a decade in average with a lot of risks. If I would like to put the cost structure to it, just to put an emphasis of what is the level of investment that current seed companies are engaged with when they want to introduce a new trait into the market, then I've written down here that it costs over $30 million to introduce a new genetically engineered trait into the marketplace. Actually, this is a very modest estimation. The figures that the industry are talking about are anywhere between 70 million to over 120 million just to introduce a single variety to the market to have a new trait. Now, let's think about a time compression event. So if I have a gene now, in the current paradigm, it takes me a decade to introduce it into a single variety. But with Moflora, how much time it takes for a new trait to be introduced commercially into a limitless number of crops, into any variety, well, it's actually just going to be the regulation time. It's just going to be a couple of years. Now, this is really a time compression effect to the market. Of course, a very interesting aspect of this technology is the regulation. Moflora has put a tremendous amount of effort in studying all the aspects of regulation, this new and innovative technology, especially concerning to its use, to its human consumption, and biosafety elements. By now, I can share with you that we are fully supported and funded in our regulation efforts by the USDA, who have identified this technology as a promising emerging technology, which is going to be beneficial to the US-based farming industry. And a letter of recommendation has been submitted to APHIS to fully waive the deregulation of this technology as GMO because we are not changing the genetic makeup of the plants. I'm going to share with you briefly what the result that we have come out looks like when we actually compare them to genetically engineered or standard GMO approach. So first of all, in both cases, you have to deliver the gene all the way to the cell. So this is not like a topical application. You really have to make sure the gene makes it all the way into the cell for the process to start, to have the gene being identified by the cell and being transcribed to RNA and then to protein. So in that regards, there's no difference. But actually, in GMO, this introduction is done not to a plant. Nobody's engineering a plant, and nobody's engineering even a seedling or a seed. In the industry, what you engineer today is actually cell culture. There is an in vitro phase, which you have to go through. And if you're successful in having a successful transformation in that cell culture, then you start to grow the plant out of it. Now, this is quite a lengthy process in you know, just rough estimates and of time and effort. It costs anywhere between a couple of $1,000 to more than that. And it's a process that takes anywhere between 6 to 18 months just to produce the first handful of plants, transformed plants, that can then, in the second generation, give you the transgenic seeds that you can start your pilot with. This process also has side effects, because what you're doing is actually you're inserting a foreign DNA into a chromosome, and you often alter the original function of the chromosome. So what the genetic engineers are actually facing is a multiple trial and error development program where they want to introduce one trait into a plant, and then they need to eliminate all the transformants which are being compromised by showing altered traits which are non-desired. That's, again, a result of this entire in vitro phase, which in Moflora, we just don't need that phase, because we don't need to change the genetic makeup, so we don't need to go and change that cell. We just, you know, take our active ingredient or supplement and apply it into the already mature and ready to market seeds. And as I mentioned, we don't have a genome integration in our case, which actually can be understood by the fact that also in nature, plant viruses do not change the genetic makeup of the plants that they attack. And by the same reason, we can understand why there is no transfer to the next generation. Of course, in genetic engineering, everything is about making a stable transformation that will be passed down to future generation. When we are genetically engineering a breed, we are creating a new breed. This is not the case in Moflora. There is no pass down to the next generation. And actually, we believe that this phenomena is attributed to a survival mechanism within the plant to protect it from epidemic spread of viruses, because what we see is that when a virus comes and there is an epidemic, you would really lose sometimes all of your crops just for that particular season. Usually, if there are any seeds from leftover surviving plants, the virus will not transfer to the next generation of plants. So it's a way, I guess, for nature to survival of epidemic attacks. There are some viruses who manage to be transferred to next generation, but most of them don't. In our case, the trade-up technology is also not being passed down to the future generation. We do not really change each and every cell in the plant that we treat. You can actually think of what is known as a mosaic-like spreading of the technology and the DNA within the plants. Interestingly enough, it even doesn't reach the pollen cells, which is very important, because pollen is one of the major attributes to the spreading of genes into the environment. And when we think about environment and we actually look at other ways to potentially unintentionally spread the DNA into the environment, and we've proved that unlike genetic engineering, in our case, the DNA that we'd like to introduce remains fully contained within the treated plant. We have even tested scenarios such as insect transmissibility, transmissibility through soil residues, transmissibility through just plant-to-plant interaction, treated to non-treated, to see if there's any exchange, or even if you use some mechanical transfer by, say, using a scissors to treat or to cut off a treated plant and then use the same scissors, maybe the sap to the next plant will transfer the trait up, and we've disproved those cases. So we can say that according to the relatively small-scale experiment that we did, we can say that the technology remains fully contained within treated plants. And this is a major issue, because if we look at the environmental concerns associated with the use of genetic engineering, whether they are, I would say, appropriately perceived concerns or over concerns which are beyond the actual, I would say, scientific concern. We know there is a lot of resistance to GMO because of environmental concerns. The first is cross-pollination. And of course, there are cases when there is a cross-pollination of genetically modified crops, sometimes even the specific trait that has been transgenically introduced is now confined in the next adjacent field as a matter of cross-pollination. And of course, the issue of wild-type cross-pollination can be more alarming. And I think there is now an evidence that in North Carolina, I think, there is now a wild weed that has been cross-pollinated by an herbicide-tolerant genetically modified crop. So now we have a wild weed which is herbicide-tolerant, and that could be a threat. Seed spill and leftover, another way of unintentionally spreading DNA to the environment because of the use of genetic engineering, and insect transmissibility. So all those issues and the concerns are alleviated by our technology, and I think it can make a difference. As any new technology, we have to further examine it, we have to further study it, we have to take this into scrutiny by regulatory agencies. We also have to make a very open and direct dialogue with the public and with consumers who are going to be eventually feeding with new products based on the use of this technology. And we think that the issues, or the main issues behind this technology, which is that the fact that we don't change the genetic makeup of the plants and we negate the issues of environmental concerns, will make a positive impact in favorably regulating this technology as non-GMO and a wide acceptance of the public to consume it, even in fresh produce such as vegetables. Now, how does it really look like when it works? So what you see here is actually 2 trays of the same variety of tomato. It is a specific variety which is susceptible to the Fusarium crown rot fungi. And what you see here scientifically can be referred to as the same genotype, the same gene of the plants to the left and the right-hand side, but it's a different phenotype. You can see a different phenomena on the right trait. And of course, this new phenomena is a new trait that has been introduced by adding the contact of this saccade to the seed treatment priming protocol of the right-hand side tomato seeds. Actually, both of them have been treated with a priming-based protocol, but only to the right-hand side we've added our genetic ingredient. And what you can see is a clear proof that the DNA that we have introduced through the seeds has actually been transformed into the plant into a protein. This protein by itself actually is responsible for a very effective control of the signs of this Fusarium. This test, by the way, has not been done internally in Moflora. We have treated the seeds, but we have sent this as an experiment to the laboratory of Professor Jacob Katan, a well-known phytopathologist and a member of the a fellow of the American Phytopathological Society, who is an expert in fungi. And we asked him to run the experiment, and when he sent us back the picture, we were very pleased with the performance. And as you could probably understand, if we are taking the seeds from the tomatoes growing on the right-hand side and then we run the test again, the result that we get are not what you see here, but what you see on the left-hand side, right? Because if I take the seeds coming from the right-hand side, the trait of the Fusarium resistance is not inherited, and I'll run the test again, and the seeds will be susceptible to the disease. We have reiterated this trial in numerous vegetables and actually numerous diseases, including rhizoctonia, including botrytis. And it's a very interesting trial. Actually, Just to elaborate a little bit more about the scientific background behind this experiment, this is actually not a single gene that we have introduced, but actually 4 different genes who work together as an operand, meaning that each of those genes is being transcribed to RNA, which produce a protein. The sufficient level of expression of those 4 different proteins take a tryptophan and convert it into PRN, which is a metabolic process in the plant. So we've actually induced the plant to go into a metabolic process through the introduction of 4 different genes. Also, the size of the insert of DNA is over 6 kilo of base pairs, which is quite a, I would say, sizable portion of DNA. It really stands to illustrate, I would say, the capacity and the potential of the technology. By the way, the source of the genetic material that has been brought into these tomato lines due to our trade-up technology does not come from vegetables. It actually comes from a bacteria, from Pseudomonas. So we can even introduce beneficial genes from bacteria into plants. Before I conclude, I would like to share with you how Moflora plans to enable the global farming communities to benefit from its technology. So our vision is, of course, to globally increase the production of agricultural crops in a sustainable manner. This is, of course, very, I would say, environmentally friendly. And we have to do it in a manner which we believe that this technology should be accessible globally. And to do so, we ask ourselves, how can we be of service? So what can we do to help the current industry to take advantage of the potential that this technology unfolds? So what we did is actually first we went and spoke to those guys on the left, all the guys which are involved in genetic research, in identifying useful genes. And they said, if we could have this technology, we could probably speed up our screening processes, which is now reliant on genetic transformation. If you could take our DNA and treat seeds and send us the seeds back ready to phenotyping, we could save years of the process. So we're actually now engaged in a quite interesting pilot with one of the largest gene discovery operation. And that's one, I would say, aspect of where this technology can potentially open a major bottleneck and unleash the potential of beneficial genes discovery. On the right-hand side, of course, we went and spoke and had a very in-depth conversation with seed companies and with seed treatment companies. And what they said was very simple. They said, we have our own processes, we have our own protocols. If your technology can fit in, we'll start using it tomorrow morning. Because actually, just a word about the seed treatment industry. Although this is a relatively young industry, maybe 10 or 15 years old, the annual turnover for this industry is now exceeding an estimate of 3 billion euro. By now, almost every seed that the farmer received is being treated either by the manufacturer or by a local distributor. As I mentioned, it's either priming protocols for the vegetables and any treatments such as coating, dressing, and palleting for the row crops and commodities. And it is Moflora's goal to seamlessly integrate its technology in the current protocol and processes. And right now, we are piloting this technology with several of the leading global players in the seed and seed treatment industry to really see how this seamlessly integrates in their technology. And I've shown you a very successful demonstration of using this technology in fungi control. Under our pilot program with selected global partners, this technology will be put to the test over the next 18 months to introduce a large variety of beneficial genes, including genes for virus control, genes for nitrogen fixation, drought tolerant, herbicide resistance, insect control, yield increase, and many other genes. As a company, as a startup company, we have very limited resources. We have tried only a handful of genes, and some of the results were really outstanding. We are really hopeful that the pilot, which is now being conducted by global partners, will be as successful, and we can all be encouraged if the results will come out that many of the beneficial genes that are today available in the industry can be more rapidly introduced into the farming community through the use of active ingredients to the seed treatment processes. And with this, I'd like to thank you for listening to the presentation. Thank you.